time to change the hayflick effect to create a longevity. Remember, a gene is just a string of amino acids that code for protein. If we can create a longevity gene that's actually only a few codons long, it's still a gene. Dr. Hayflick published an article where it was noticed that any kind of sites from any kind of tissue would divide only a certain number of times. This was called the hayflick effect. It's possible that doubling the number of times a site can divide, or even making it be able to divide 10 times as many times, would cause greater longevity, as well as greater youthfulness of tissues. Now, there's an important difference between a tissue that just replicates from a tissue that divides normally. A tissue that just replicates is sometimes considered cancerous, whereas a tissue with well-being that's just able to divide more times would create a longer period of youthfulness. One of the ways to create a human longevity gene that causes the hayflick effect to diminish or permit sites to divide 10 times as many times prior to ceasing goes like this. Just take a normal human tissue culture, then give that tissue culture mutagens that cause mutations at the DNA of the tissue culture. Then, every amount of time that is half the usual amount of time that a tissue takes to divide, introduce a stable isotope marker. Thus, as the tissue divides, going through each hayflick division, the material will accumulate perhaps 40 or 100 different marker isotopes. With the presence of the mutagen, some of the materials may have their genetics changed so that their hayflick effect is changed. Thus, after 99% of all the tissues have ceased dividing, make a contact copy culture of the human tissue that remains. Transferred to fresh medium, see if any of these possible mutants continue dividing beyond the previous hayflick effect. Then you will have found human tissue lines that are functional, yet have surpassed their previous hayflick effect, as proven with the isotope labeling. Now, a word about the mutagen used to create this effect. When you think about it, a mutagen just creates a mutation, and there's a wide variety. There's everything from things like UV radiation that change single codons to things like chromosome tip overlap change, which regroup entire sets of genes simultaneously. There's also colchicine, which really doubles the amount of genetic material. What's the cleverest kind of mutation to create that is most likely to produce tissues that continue to divide, removing the hayflick effect? I think that mutagens that cause the rotational swap out of anything from 300 to 700 or possibly 7,000 codons is likely to create the recombinations that are most likely to change the hayflick effect such that tissues can divide 10 times as long, absent senescence. One approach to that would be creating or using what I believe may be published variants on transfer RNA that are responsive to light. Because as the transfer RNA cause the duplication of the entire genome, you can use light to interrupt them once every 300, 700, or 7,000 codons, which would cause the codons to possibly shift their position. Another possibility is the use of brief bursts of ultraviolet light or spatially patterned ultraviolet light. There's a researcher that I read about at Technology Review that was able to get optical microscopy down to less than 100 nanometers, and they believe that it could occur between 10 and 20 nanometers using what are known as evanescent waves. Now, if you create a little striped pattern of ultraviolet radiation that has a width of 10 or 20 nanometers, you'll be able to create little chunks of divided DNA that will then recombine somewhat randomly, similar to the tips of chromosomes swapping. Thus, you can use a particularly striated spatial form of ultraviolet radiation to create new patterns of mutagenesis, which you can use to change the hayflick effect, to create a new human gene, which is 10 times as resistant to senescence. Then, try it out on mice. See if the mice with the new human gene that basically removes the hayflick effect causes the mice to live longer. If it works for them, then try it on human beings to create much longer human longevity.